I would like to talk about the pre-existing T cells, and I noticed that the title of this conference has changed, and nowadays you are talking about mastering immunity, not only immunogenicity. This is the reason why I adapt myself as the immune response, and I would like to talk about the specificity and phenotype of pre-existing T cell, raised against therapeutic proteins, but also against vaccine candidates and in healthy donors. So, the issue that interests me is that we are developing many therapeutic proteins and many vaccine candidates, and we wish to know whether they can induce immune response, T cells and antibody response in the patients before any injection in the patients. And for this reason, we look for technologies of immunogenicity prediction, especially because we know that the animal models are not really informative. So we need alternatives. And the alternative is the LC donors. And we try to get as much as possible information from the LC donors. I do not see the pointer. Okay, yeah. Information from the LC donors who are expected to have naive cells to account for the memory response that may occur in the patients. So that means that we are looking for footprints from the memory response in the naive cells, and we know that these footprints these footprints may belong either to unwanted immunogenicity for therapeutic proteins, or, in contrast, to a very nice immune response for a vaccine candidate. So this is what we are doing and what we wish to do. So let's go for the specificity of the pre-existing T cells. I like very much to remind you this very nice paper from Jenkins' lab. And they use these very amazing tools that you can get also from ProImmune, is a tetramer. So they use a tetramer specific for different epitopes in mice, and these tetramers allow them to track the specific T cells. And so they did these experiments using naive mice, and they tracked in the complete spleen of the mice, and you see that there are very few cells. But the number of cells is very variable. It depends on the epitope. First message. The second one is that when they immunize the mice, they get very strong response, in this case, here or here. And you can see that the amplitude of the T cell response is related to the number of epitope-specific naive T cells. That means that higher the response is, it was due to the high number of precursors at the beginning of the response. So there is really a relationship between the memory response and the naive response. Anyway, it's just mice. What about humans? I also like very much this paper from Bill Koch laboratories. They also use tetramers, and they have investigate the response of three epitopes specific for the protective antigen of Bacillus anthracis. It's DR1 restricted, and they use three tetramers. And first, they track the specific cells in the non-vaccinated donors. And you see here the number of cells. It's a log scale here. It's very variable from one epitope to another one. And you can see this peptide is at very low frequencies in contrast to that. But you know, it's very low frequent cells. It's around 1 cell per million of cells. And when they look the same population in the vaccinees, you see the number is largely higher. In this case, they found exactly the same profile, and but just the amplitude has been modified. And so again, you see that higher is the response depending on the number of precursors at the beginning. So you see that it could be very informative to get data from the naive donors. But there are not so many technologies to quantify the number of cells in naive donors. There are technology to detect them, but to quantify they are less they are not so multiple. The first one is the tetramer technology, as I show you. The issue with this approach is that finally it's very appropriate to evaluate the response epitope per epitope. But if you wish to do it for a whole protein, it's much more difficult. Because you need to know the peptide, you need to know the HLA molecule, so it's not completely convenient. Another possibility is to use the technology developed by Federica Sallusto, but finally it has been applied to high immunogens only, and they cannot detect for low immunogens. And the last one is what we did in my lab is what we call the T-cell amplification assays that I will detail in the next slide. So the idea is that there are 3 steps, and we mix the donor cells previously loaded with the antigen with the purified CD4 cells, and this is done in multiple replicates, and we stimulate the cells several times with dendritic cell loaded with the antigens, and at the end we perform a specificity assay using ELISpot. And you can see that using molecules such as ovalbumin, not all the wells are positive, only part of them. And so you see the spots here in the red bars, here no response. Also, all the wells have been treated exactly in the same manner. And this is due simply because at the initiation of the culture, the frequency is so low that we are in limiting dilution conditions. And we can use these conditions, the limiting dilution conditions, to access to cell quantification, considering the distribution follow Poisson's distribution. And this is what we have done a couple of years ago, so now it's an old paper, and we have applied this approach to therapeutic antibodies. And to make the story very short, you see that for high immunogenic molecules like the murine antibody or the KLH, we find around they are higher than 1 cell per million of cell. And this is, in my lab, all the molecules with this kind of response are known to be immunogenic. And so you see for the murine antibodies around 2 cells per million, KLH is 30. And for low immunogenic antibodies fusion proteins, etanercept or trastuzumab, they are below the range of 0.1 cells per million. And for the immunogenic antibodies, like adalimumab, rituximab, and infliximab, they are all in the range of 0.1 to 1 cell per million of cells. There is only one exception, is bevacizumab. It has been given in cancer patients only, and we suspect that it was the reason why it was found non-immunogenic. Anyway, by this way, you see that we can rank the molecule based on the immunogenicity level. In the frame of ABIRISK, we continue to work with these antibodies, and we have definitively mapped the T-cell epitope of these antibodies. I will show you for Ritux and Inflix, and So we have applied exactly the same strategy, but at the end we have screened the cells with overlapping peptides. And this is exactly this kind of data we get, and you see from the first ELISpot we have a response to Retux to one pool of peptides and finally to one peptide, and for Enflix is the same. At the end we get 9 epitopes for rituximab and 9 epitopes for infliximab. But we had also access to patients that have developed antidrug antibodies. And so for this patient, we applied a different strategy as we were interested in the memory response, not the naive response. We have made a very short-term T cell assay, or a very short stimulation phase. And then we prime with the peptide that we have already identified the PBMCs of these donors, and we screen for IL-5 ELISpot. The idea is to see whether the peptide we identified in the LC donors are indeed relevant for the response in the ADA patients. It doesn't mean that other epitopes maybe are also present, but we do not detect them as we do not screen for all the peptides, just to see whether the peptides are relevant. And when we made the comparison, you can see that we get the response in many cases, and showing that indeed the peptides we identified in the LC or participate to the immune response in the anti-drug antibody patients. Come back to vaccine. I also have an example of that for a vaccine. A couple of years ago, I participated to the HIV network in France, and I'm still part of the Labo of Excellence of the Vaccine Research Institute. And we were interested by a peptide vaccine comprising 5 long fragments. And these peptides have been initially designed based on the CD8 epitopes, but not at all with the CD4 epitopes. But they found that the vaccine induced also the CD4, and this is why we investigate in more details the CD4 T cell epitopes in these vaccine, synthetic vaccine. So finally, we took advantage of the large clinical trials that have been done on these peptides to confront our technology of immunogenicity prediction. So we submit the peptides to the different technology of the lab. And the first one was the binding assays. And so we have a collection of HLA-DR molecule, and you see that the 5 fragments, they can be active to bind to different HLA-DR molecule. Especially 3 peptides bind to multiple HLA-DR molecules, and 1 G2 and P1. But when we submit this peptide to the T cell amplification assay using multiple donors, seronegative, no, it's LC donors again, you see that one peptide emerge as the most T cell stimulating. Don't forget it's a log scale. And so clearly the G2 is much more stimulating than the others, and then the P1, and the other one, it was clearly less. At the end, we derive more than 600 different specific T cell lines, and we can present these data in a different way. You see the distribution of the T cell lines based on the specificity. Clearly, again, the G2 is much more potent and much more stronger than the others. And when we compare with the vaccine tree, or the 132 volunteers have been immunized with this vaccine, if you look at the distribution of the responding donors, I know it's not exactly the same distribution, but anyway, you can really see that the G2 is really much more T cell stimulating than the other. So again, it means that it's different distribution, it's different way, but we will conclude that this peptide is probably much more interesting than the others, and indeed, this is what was found in the clinical trial. Okay, so now I will talk about the phenotypes. Because generally we believe that before any injection, the cells are naive. But I will show you that it's not systematically the case. This is a paper we published 2 years ago in GI. In my lab, we are also very interested in tumor antigens. And we especially are interested in proteins that are transiently expressed in normal tissue. And this is the case of cyclin B1, but are expressed in a high level in the tumor cells. So I would say I have a sort of opportunistic behavior, because sometimes I notice that one target has been already investigated for the CD8, but not for the CD4. And as we are very good at the CD4, but not really for the CD8, so I am very interested by this kind of target. So they already been proved to be relevant, as CD8 CTL have been demonstrated to cyclin B1, and so we investigate the CD4 response. And we applied again the T-cell amplification assay. And again, when we do this with cyclin B1, we find a very good frequency of CD4 cells. You see around 1 cell per million of cells. We find that they have both naive and memory phenotype again. And finally, we identify more than 20 different CD4 T-cell epitopes in this protein. It's a very huge protein. You see the number of T-cell lines we derive. Sometimes we derive more than 50 T-cell lines for the same peptides, and some peptides are very common to multiple donors. For this one, for instance, or this one. So when we did this work, we were very happy, and we asked my colleague from the European Hospital, Georges Pompidou, Eric Tartour, to investigate the memory response in patients, in cancer patients, to see whether they have already a sort of immunity against these proteins. And so he applied exactly the same protocol that we did for the therapeutic proteins. And it's a very short-term T-cell assays to track the memory response. And this is the mean of 9 donors for the lung and 5 for the head and neck. And you can see that part of the T-cell epitope, but not all, contribute to a response, in spontaneous response in these patients. But our surprise is to see in the control that we also get exactly the same profile of response in the LC donors, which means that there are some T cells already expanded in the LC donors against cyclin B1. So if there are many T cells, maybe there are many antibodies, and we screen for antibodies against cyclin B1 in the LC donors. And indeed, we find many antibodies against cyclin B1 in all the donors. So we screen more than 100 serums, and they almost are all positive in contrast, in comparison to a control antigen. But when we screen for the cancer patients, we do not find any difference between LC and cancer patients. The level of antibody is the same. So we have the feeling that there is a loss of tolerance to cyclin B1, whatever the donor, LC or cancer, and we suspect it is due to the expansion of CD4 cells that are specific to part of the T cell epitope only. So to conclude my talk, I hope I have convinced you that a part of the answer about the memory response is imprinted in the naive repertoire of the LC donors. It makes a rationale for immunogenicity prediction to rank immunogenic proteins, to identify immunogenic peptides. And I also hope to convince you that the pre-existing cells are not necessarily naive cells. And for instance, we find for factor VIII and for cyclin B1 a high frequency of cells, demonstrating the escape from the central tolerance. But there is a great difference between these two antigens. One, we do not detect any expansion for the factor VIII specific cells, in contrast to the cyclin B1, and it was followed by antibody response. I will be pleased to acknowledge the people who did the work, people in my lab, but also people at INSERM. You know, it's a French Governmental, it's equivalent to NIH, but the French version. It's largely smaller. And my friends Sebastian and Eric, and we also collaborate with Novartis for the MAPS assay. Thank you very much for your attention.